Hello everyone, and welcome to today's The Scientist webinar. I'm Betsy Young, the technical editor for The Scientist, and I'll be moderating our discussion. Today, our panel of experts will discuss emerging technologies for disease modeling in regenerative medicine and stem cell therapy. We like our webinars to be interactive. We encourage you to send us your questions or comments at any point during the webinar, and the panelists will address these during the Q&A session following the presentations. To ask a question, simply type your query into the question box located on the bottom left-hand side of your screen. We will try to address as many of these questions as we can during the Q&A session. The webinar platform is user-friendly. You can move or resize any of the windows by simply grabbing them at the top or stretching them at the bottom right-hand corner. You may need to move or minimize some of the windows to see the live view. The webinar will be archived on the Scientist website, and we will send you the link via email within a couple of days. Please note that you will not be able to download the presentation slides. I'd like to take this opportunity to thank our webinar sponsors. Rockland Immunochemicals offers each academic, biopharma, and diagnostic professional thousands of antibodies with the aim of providing the right antibody that is the perfect fit for every occasion. With facilities in Pennsylvania for over 50 years, Rockland manufactures products ideally suited for integration into critical assays such as Western blotting, immunohistochemistry, immunofluorescence microscopy, ELISA, and flow cytometry. No matter the context, basic research to disease therapy, phosphorylated to methylated and beyond, Rockland embraces the challenge to design, produce, validate, and deliver the best antibodies and life science reagents available in the market today. With Rockland, your solution is complete. Baker Ruskin has been leading a revolution in advanced cell research for more than 20 years, working with pioneering scientists around the world to mimic in vivo physiology for more repeatable, reliable results. Its advanced lines of controlled oxygen workstations and media conditioning solutions allows you to study the most complex of cell interactions under perfect physiological normoxia, tightly regulating and precisely maintaining oxygen, carbon dioxide, temperature, and humidity. To learn how Baker Ruskin products can benefit your research, visit bakerruskin.com. Our sponsors have provided us with some helpful resources, and we've posted these in our resource list located on the left side of the screen. You can access and download these documents at any time during the webinar. And with that, let me introduce our first speaker, Dr. Yeon Kim. Dr. Kim is an assistant professor of neurology at Harvard Medical School and an assistant in neurology in the Genetics and Aging Research Unit at Massachusetts General Hospital. He received his PhD at Korea Advanced Institute of Science and Technology in South Korea. He started his postgraduate work at the Brain Disease Research Center at Aju University Medical School in South Korea and completed his postdoctoral training in Dr. Dora M. Kovac's laboratory at Massachusetts General Hospital, where he was appointed as an assistant professor in 2009. Dr. Kim has been investigating the physiological and pathological functions of Alzheimer's disease secretases, which have been one of the major drug targets for clinical trials. Recently, Dr. Kim's laboratory also worked on establishing human neural cell culture models of Alzheimer's disease, which can be applied to screen Alzheimer's disease drug candidates. By adopting a novel three-dimensional cell culture system, Dr. Kim's team, together with Dr. Tanzi's lab, has recently shown that familial Alzheimer's disease mutations could induce robust beta-amyloid and tau pathologies in human neural cell cultures for the first time. Dr. Kim?
Thank you very much for the invitation and nice introduction. Today, I'm going to present our recent studies on a three-dimensional human neural cell culture model of Alzheimer's disease. At the beginning of this presentation, I'll give a brief overview of Alzheimer's disease, then explain why we developed the 3D culture models of AD. Next, I will explain why and how we switched from conventional 2D culture models into the 3D system to recapitulate robust beta amyloid and tau pathology. At the end of this talk, I'll also discuss about the challenges and prospects of 3D culture system in modeling Alzheimer's disease and possibly other neurodegenerative diseases. Okay, now Alzheimer's disease is 6th leading cause of death in the United States, and the number of patients, currently more than 5 million, are expected to be increased in next decade. However, there is no cure at this moment. Alzheimer's disease patients suffer gradual loss of memory and cognitive function. In the pathological point of view, Alzheimer's disease is defined by two major hallmarks, which are the beta amyloid plaques and neurofibrillary tangles. As you can see in the right diagram, beta amyloid plaque is extracellular aggregates which are made from very small 4 kilodalton peptide called A-beta, while neurofibrillary tangle is an intracellular aggregate composed mainly of hyperphosphorylated tau protein. So A-beta peptides are generated from the sequential cleavage of amyloid beta precursor protein, which is APP. Studies showed that this sequential cleavage is mediated by two secretases, beta secretase and gamma secretase. And as you can see here, that these cleavage leads to the generation of A-beta peptide monomers, which is 4 kilodalton. And then it leads to the aggregation to make a most pathogenic species, which is soluble A-beta oligomers, and also A-beta fibrils and A-beta plaques. For neurofibrillary tangles, NFT is made from aggregated hyperphosphorylated tau protein. Tau is microtubule binding protein. In disease conditions, tau proteins are hyperphosphorylated, released from the axon microtubules, accumulated in abnormal compartments like dendrites and soma, and aggregate into unique fibrillary structure, including PHF, so paired helical filaments, and then finally leads to the tangles. So next, the immediate question is how beta amyloid plaque and NFT contribute to pathogenesis of AD. The beta amyloid cascade hypothesis of Alzheimer's has been reigning for the last 2 decades. A-beta hypothesis suggests that excess accumulation of pathogenic A-beta species, either by familiar Alzheimer's disease mutation in APP or presenilin, or by decrease in A-beta clearance, triggers vicious toxic cascade, including synaptic deficits, microglial activation, neural injuries, and hyperphosphorylation and aggregation of tau protein, and in the end, eventually leads to the neuronal death. Many genetic and biochemical evidences support this hypothesis, and therefore, the majority of clinical trials of AD are based on this hypothesis. However, interestingly, no single in vitro or in vivo model have been able to fully recapitulate both beta amyloid plaques nor the neurofibrillary tangles derived from APP or presenilin FAD mutation. This point is very clear if we are looking at the most popular Alzheimer's disease transgenic mouse models. And these are the standard AD model for basic mechanistic studies and preclinical AD drug trials. And this model, harbor single or multiple familiar Alzheimer's disease mutations and recapitulate symbolic AD pathologies, including beta amyloid plaque, cognitive impairment, synaptic and neural injuries, microglia activation, and even neuronal loss in some models. However, no clear neurofibrillary tangle nor neurodegeneration observed in all of these models. So according to the beta amyloid cascade hypothesis, excess accumulation of A-beta peptide would lead to the tangle and neurodegeneration. So does this mean that something is missing in current A-beta cascade hypothesis? Another possible explanation is because from the species-specific differences between mouse and human. Indeed, adult mouse does not express 6 different human tau isoforms, which is very important for the tau tangle pathologies. And even the studies shown that endogenous mouse tau protein seems to interfere the aggregation of human tau protein. So if that is the case, can you see tau tangles in human neuronal models? So advances in stem cell technology make it possible to generate adult neurons from the fibroblasts prepared from Alzheimer's disease patients. So this model, in theory, can provide an ideal human model system to avoid any potential species-specific differences, and of course, the tau should be the human tau forms. So these are summary table of currently generated human neurons harboring either preserine or APP familiar Alzheimer's disease mutations. As predicted, most of these neurons with the FAD mutation showed significant increases in pathogenic A-beta 42 levels or ratio due to the altered cleavage of amyloid beta precursor protein. Interestingly, some of FAD neurons, especially with APP duplication or London FAD mutation, showed a significant increase in both total and phospho-tau levels, which may suggest the early signs of neurofibrillary tangles. But still, there's no robust beta amyloid plaques nor neurofibrillary tangles in human FAD neurons. One possible reason for not being able to see these both robust pathologs is because the pathogenic A-beta levels are too low in this model. If you are looking back these previous slides, and you can see that the A-beta 42 levels are around the 4 to 10 femtomolar per milligram, and some of models it's not even detected. In human Alzheimer's disease patients, the average levels of A-beta 42 in the brain is 5 to 10 picomolar per milligram. So it's almost like 1,000-fold differences in terms of levels. And then one more thing I want to point out is that these iPSC-derived neurons, still it's nicely shown a lot of nice adult feature of neurons, but still show some of the embryonic characters. Especially the mature tau isoform levels are very low in differentiated human neurons. So we make a goal. So we wanted to create human neural stem cell models producing high levels of pathogenic A-beta peptides, which are comparable to human AD brains. And also, we want to generate it 2D or 3D. Actually, we are starting from the 2D system. That's a conventional system. And then we switch to 3D. I'll explain later why we did it, and to recapitulate both beta amyloid plaque and neurofibrillary tangles. Today, most of the data I'm showing today is already published, and then I'll make a little bit faster some of the details. I hope you can take a look at our original publications. Okay, this is the experimental design, and then you can see we chose immortalized human neural progenitor cell line, which is called RANCell VM. So these cell lines are derived from human fetal brains, immortalized, differentiated into 70% neurons, 30% astrocytes, and very small amount of oligodendrocytes. And it is already reported that it shows that the voltage-gated potassium channel and sodium channels. And then also, this is most important, it can be maintained up to 45 PASI numbers without any deterioration of the differentiability or survival rate, which is very important for us because we want to create some kind of cell lines that can be maintained. And then next we used the lentiviral vector system to transfect these cells to express high levels of FAD genes with the multiple mutations. And then after that, we use the FACS sorting to not only cut out the non-transfected cells, we also enrich the cells which express high levels of these FAD genes. And we use the two different 3D culture systems. I'll explain later why we had to go with these two systems, but generally it takes about 6 weeks for the differentiation and development of A-beta plaques. And then we see the tau tangle pathology around 8 to 10 weeks. Okay, this is the diagram showing lentiviral vector. So this is polycistronic vector, which you can see IRES system. So the nice thing about this, you can see that, for example, APP in the middle, you can see these APP, the Swedish and London mutation was linked to the GFP. So let's say we are de facto sorting high levels of GFP expressing cells. That means we are selecting cells with high levels of APP with mutations. And same thing for the presenilin, and even we can make it combined together. And these are the summary of cell lines we published. Right now we have more than the 41 cell lines in total, but these 6 cell line, FA cell line, are major cell line we published at the beginning. So this is one of the examples how we did the FACS sorting. So basically it's simple. We can transfect with the lentiviral vector, and as I just briefly mentioned, that APP is linked to the GFP levels in here. And you can see we selected top 2%, top 10%, and top 50%. So that means we have 3 different lines with different expression levels of A-beta. And as a control, we sorted same signals with the GFP alone. That is actually good control for our studies. So this is one of the proofs that protein is expressed here. And you can see in the leftmost side of each gel, you can see control LAN G, GFP only, and LAN M, M cherry only. And you can see APP levels rise robustly increased in the cell line called GA, which is APP overexpressing cells. And you can also see LAN cell with MGAP, which have APP and presenilin. You can see robust increase in these cell lines. So this is the initial screening. And we characterize these cells after differentiate for 6 weeks. So using the qPCR array, we see that there are robust increase of neural markers and astrocyte markers. And we also confirmed, as previously reported, that these cells expresses the voltage-gated sodium channel and potassium channel currents. And also we see some of the synaptic markers, which is punctated form. So it looks like it has some synapse-like structure after 6 to 7 week differentiation. And then most important thing is A-beta levels. And we are happy to see that these cell lines and the HLAN MGAP is having cells with APP and presenilin, probably the most highest A-beta secreting cells in the when we do the publication that time. And you can see that A-beta 42 level is close to 4 picomole per milligram, which is very close to the average the A-beta 42 level. And this is almost 500-fold higher than iPSC-derived FAD neurons. And this Western blot shows how robust the A-beta expression in these cell lines. Generally, A-beta detection is possible by very sensitive ELISA system. But in this case, we can just culture the cells out of 2 mL of media. We take out 30 microliters, very small amount, and then just run to the gel. And just conventional Western, we can detect A-beta, these high levels. So these cells is proven to very high, robust high levels A-beta 40 and 42. And we have a big frustration. So we differentiate them in the 2D culture conditions, and which is that generally you can see the cells are attached to the bottom of the dish. And then with the growth factor deprivation, cells start differentiate. And what we found is that after 6 to 12-week differentiation, we see a lot of A-beta accumulates in the media of these cells, but we do not see any aggregation of A-beta peptide, nor any increase of phospho-tau or tau aggregations. So it was very frustrating. But at that point, we thought, maybe it's not just the cell line problems. Maybe it's a system problems. You can easily imagine that every 3 days we are changing media. So which means that A-beta is generally secreted out to the media, and then we take it away. So we are actually get rid of A-beta every 3 days. And that's probably why A-beta cannot aggregate inside this system. So what we thought about it, what if we are growing them in the gels? And these gels is actually make them protecting them from the diffusion of A-beta. At the same time, with the connections, they can actually achieve the high concentration A-beta, local concentration that is enough to make aggregation. For this, we use a matrix gel. This is a very well-known gel system for the stem cell culture. So basically, it's the most laminin collagen. And nice thing about this is it liquefied at 4 degrees. So we are actually mixing them together with the cells 4 degrees, and then put it onto the room temperature, and then we can get cells in 3D. And after that, we start differentiating. So this is why we make, I can explain why we made 2 different 3D culture systems. Because one of the 3D culture problem is that because it's very thick, so it's not that easy to measure what's happening in the middle of the gels. Especially there are limitations, confocal and others. So we want to make some system we can easily do immunostaining, immunohistochemistry, and something we have enough amount for the biochemical studies. So that's why we made a thin layer culture fit into the 96L, 384L, and other formats. It's about 200, 300 micrometer. We have a 2 to 3 layered cells in here. And thick layer culture is actually big, and then they are almost close to the size of mouse brain. And then they can differentiate very well in the 24-well or 6-well dishes. And we use mostly biochemical analysis, but we can actually now start doing paraffin section of the thick layer cultures. And this is one of the example of 3D culture differentiated cells, and I'll just skip it. So we see the same thing. We found that there are robust increase of neuronal cells, about 70% it matches, and then about 30% of them differentiate in astrocytes. And we could not detect very well the oligodendrocyte. And one of the advantages of these 3D culture, actually I think cells are more stable in the 3D as compared to 2D. This is actually cultures from the 6 months. And you can appreciate they are making nice neurites and all the connections even after this long time. You know, one of the technical challenge of the iPSC culture in 2D, because those matrix coating are detaching after 12 weeks, 9 to 12 weeks. The cells are floats away. But in this system, cells are in the huge amount of gel, so they stay very long time. So our record is 6 months, but I think it's a matter of like whether there's any contamination or others. Otherwise, it's very stable. And this is one of the first staining we did with A-beta. So the red is the A-beta antibody, very specifically recognizing A-beta peptide and plaques. And then the green is GFP, which is expressed in all of these cell lines. So you can see that there's nothing red in the control NG, which is left side pictures. And then when you see in the middle picture, which is GA, it's only APP in London. This says high levels A-beta, but mostly A-beta 40. So they are not very aggregating species. So you can see a very small spots here. But when you go to MGAF cell line, which has the pristin and APP together, we see that there are the diffuse aggregation of these A-beta peptide, and we also see some of very tight aggregates in the cultures. So, okay, so these are the G sections of these tight, one of the tight aggregates. You can appreciate those red aggregates from the bottom to the top. So our approximation is about these particular aggregates is about 50 micrometer. We are actually matching with the average A-beta plaque size in patient brains. And then we turn to the biochemical staining because histochemical staining because fluorescence always can give some artifacts. So we use the conventional classical depth staining, and you can appreciate these control cells. You can see those blue are the nuclei hook staining, and the brown is A beta antibody with depth staining. You can appreciate these two AD lines show robust increases of these depth staining, the tight and diffuse aggregates. In high resolution picture, you can appreciate definitely these aggregates is not inside the cell. It generates from the outside. Also, inhibitor of A beta by beta secretase inhibitor, gamma secretase inhibitor completely block the developing these brown signals. Okay, this is one of the pictures from the paraffin section. So thick layer culture, we also see the A beta aggregates. So you can see in the bottom line, which is Neurology. So you can see a lot of connection of NAP2 urite connections, and you can see that there are A beta aggregates very close to the surface of 3D culture. Yeah, so this is another proof that the A beta aggregated in this system. So this is biochemical analysis. So in the insoluble fraction from the thick layer culture. And you can see, as expected, in the very TBS insoluble and SDS soluble fraction, you can see accumulation of A beta species. Especially, you may remember HLN cells, most highest A beta secreting cell line. And you can see that accumulation of A beta monomers in the insoluble fractions, but you can also see dimer, trimer, and tetramers. I'd like to remind you that in the media, we only see the A beta monomers. And this is another sign that the A beta species in this culture is aggregated. And even we can see SDS insoluble formic acid soluble fraction, there are A beta stays there, which is extremely aggregated species. And we checked the time course, and then we found that starting from the 2 weeks, 14 weeks, these A beta species, aggregated A beta species, is increased proportionally. So now switch to the tau pathology, which is the most exciting part of our studies. And before going into the tau, I want to show something about advantage of 3D culture, not just A beta aggregation, but in terms of recapitulating other neuronal physiology. So this is the quantitative RT-PCR analysis of neural markers, which I shown you in one of previous slides. But what we did here is that we compared the exact same cell line for 7-week differentiation in 2D versus 3D. And even in 2D, you can nicely see that neural markers increased by 15-fold, 33-fold. But if you can see the 3D differentiated condition, there are robust increase as compared to 2D in terms of all the differentiation markers. Let's say if you see the map, which is tau genes, in the 2D, there are 33-fold increases as compared to the undifferentiation, which is quite nice amount. But if you are looking at 3D differentiated, exact same conditions, and then same cell line, and you can see 525-fold, which is robust increases. And this increase of tau is not only the levels. This is the RT-PCR showing those other isoform, 4-repeat tau and 3-repeat tau. And if you are looking at in the 2D culture, in LAN-G control cells, there are increase of 4-repeat tau as compared to the undifferentiated. But if you are looking at the box in the right side, in the 3D condition, we see the ratio between 4-repeat and 3-repeat tau is robustly enhanced as compared to 2D. And the right side is other human brain controls. And other human brains, it's 1-to-1 ratio. So our 3D RAN cells actually very nicely mimicking the condition of 4-repeat and 3-repeat tau. And this data is not published, but I want to show you as a references. And we actually grow some human iPSC-derived neural precursor cells, and we differentiate them into 12 weeks. And then we also did exact same analysis. And you can see it in the 2D. We could not even detect 4-repeat tau species. 3D is make it a little bit better, but still it's very low levels, and even not smaller than 2D differentiated LAN-G cells. So that's one of the concerns I have about recapitulating tau pathways in iPSC-derived neurons, because the full repeat tau, other isoform levels are very low, and then looks like in the 3D culture condition with the RAN cells, we can actually achieve high levels of full repeat tau. Okay, this is actually first data we found about the biochemistry. So you can see the hyperphosphorylated tau we can find in the insular refractions. You can see that the ATA, the antibody against phospho-tau, shows nice increase of these species in AD cells as compared to the control NG cells. And you can see it even in the SCS insoluble formic acid soluble fractions, which is highly tightly aggregated fractions. You can see there are phospho-tau. And we have another assay with the detergent, sarcosis detergent, which also show the robust aggregation of tau species in these cells. And we did turn into the immunohistochemistry. So as you can explain before, that hematoxin is a blue, the nuclei staining. And you can see this brown, which is the antibody against phospho-tau. And it's quite interesting because you can see that these individual neurons with a high level in cell body and neurites with very dystrophic neurites forms. And also we see the area which is highly enriched with this phospho-tau. And we think that the area may be possibly highly enriched with A-beta oligomers. And we did unbiased counting as expected. There are robust increase of the hyperphosphorylated tau containing neurites and cell bodies in AD cells as compared to the control. And this is another immunofluorescence staining, but this line is actually more enriched line, similar to HRAN9 I showed you. So we see more robust tau pathologies. You can see in the left section, you can appreciate there are green. Phospho-tau staining is elevated in AD cell line as compared to the control. And we see it in the neurites, especially dendritic compartment, and then you see in the cell bodies. And also even we see cell body with the spotlight structure. We think it's a forms of pretangles, but need more the experiments to confirm it. And biochemistry show robust increase of the phospho-tau and total tau in the insoluble fractions in these AD cells. And one more, the things we tested, whether is any kinase involved with increase of phospho-tau. Among those tau kinases, GSK3 beta is well-known characterized kinases. Still, there are many possible tau kinases that mediate a beta-induced tau phosphorylation, but we actually treated our system with these GSK3 beta inhibitors, and you can appreciate the robust decrease of the phospho-tau in our system, just a 1 week of treatment. And then we turn into the immuno-EM, because one of the decisive evidence for the tau fiber is to show the structures using the electron microscopy. And from the insoluble fractions, we actually go to the EM studies with the antibody against the tau. In the right side, B, you can see those black dots. That's tau 46 antibody, which recognize tau protein. And if you are looking at these filament-like structure, you can appreciate the inside. There are some like filament-like structure which is matching with the size of tau filament previously reported. But after we publish this data, we have several criticisms whether this doesn't really look like the helical twisted PHF tau filaments. So what we thought is maybe it's because we just use 7 weeks. So maybe longer differentiation, we get more robust tau pathology, and then we have more robust tau filament structures. And indeed, that's the case. As you can see here, you can appreciate this filament is not only just straight line, you can see those twisted in each side. We calculated average the pitch size, and it's about 120 nanometer, which is very close to what is known for tau filaments from AD patient brains. So these all together strongly support that the presenilin FAD mutation and APPFA mutation can induce not only A beta aggregation, but also tau aggregation in vitro. So I'm going to turn back to the beta amyloid hypothesis. One of the interesting things we can test in our model is whether these hypotheses are really valid, because we are currently doing a lot of clinical trials is trying to block A beta accumulation. And then the goal is because A beta accumulation is very early stages, so we can actually block all the pathological changes downstream. But is it really true? So we want to test it out in our system. So this is actually one of the pictures we already shown. What we did is we treated for 3 weeks with beta amyloid inhibitors, which is beta secretase inhibitor and gamma secretase inhibitor. And we see there are robust decrease in these phospho-tau bearing neurons by blocking A beta generation. Same is true for the more robust FAD lines we have, and you can appreciate those brown is phospho-tau staining. So you can see there are robust increase of phospho-tau staining in AD cells as compared to control, and we treat 3 weeks with the beta secretase inhibitor and gamma secretase inhibitor. You can appreciate these neurite phospho-tau staining are significantly decreased. Our data from the Western blot and other biochemical also supports our conclusion. It seems that when you block the A beta generation in this model, it's robustly blocking phospho-tau levels in the in-situ refractions. So this is our conclusion. We think that our model at least supports A beta hypothesis of AD, which propose that accumulation of A beta drives tau pathway. And one of the exciting applications of our model is that this can be used as a high throughput platform for the AD drug screening, because current mouse model is actually too expensive to do those high throughput screening, but our system is way cheaper and faster than mouse model. And this is the last slide, and as you can see that we may recapitulate some of the AD pathogenesis in our cell line model, but there are many challenges. One of the major challenges is that to precisely reconstitute brain regions that are most affected in AD, so hippocampus and the cortex. And maybe HIC-PC-derived models and or even cerebral organoid model will help this reconstitute structures. And another thing is to integrate neuroinflammatory components. And as you know, that our cells cannot differentiate into the microglia cells. So this system has astrocytes and neurons, but does not have microglia component, which is based on the A beta hypothesis. Its activation of the microglia and astrocyte is very essential for the neurodegeneration. So that is another challenge for the future. And another thing is that whether we can make a recapitulate functional deficits in neurons. Because advantage of mouse model, we can actually do the behavior test to test their memories. But this system currently is just only for the levels, the biochemical levels. So I think that's another big challenge in the future. And another thing is that we are focusing a lot is towards a standard model of human cellular AD model for drug screening. So we are having a lot of progress in the single clonal screening and 3D co-culture model, and we are hoping that in the future that we can distribute some of our models as one of the models of testing AD drugs. Okay, this is the last slide. So I would like to thank all of our lab members, collaborators, and funding supports. In particular, I would like to thank Dr. Rudolf Tanji, who has been a close collaborator and main supporter for the 3D culture project. Also, I'd like to mention that continuous support from Cure Alzheimer's Fund was essential for this project. Cure Alzheimer's Fund also generally supports our current 3D high throughput drug screening efforts, and we really appreciate for their support. Thank you so much.
Thank you, Dr. Kim. As a reminder, please continue to submit your questions for the speakers in the question box at the bottom of your screen. Now I'd like to welcome our second speaker, Dr. Jason Spence. Dr. Spence is an assistant professor at the University of Michigan. His research focuses on using mouse models to study embryonic development of GI and associated organs, and on using three-dimensional cell culture models to study human development and disease. Dr. Spence received his PhD from Miami University in Ohio under the supervision of Katia Del Rio Sonis. As a research fellow in the lab of Jim Wells at Cincinnati Children's Hospital, his work focused on understanding mechanisms that regulate embryonic development of endoderm-derived tissue and utilizing human pluripotent stem cells to understand human differentiation and development. During this time, he pioneered efforts to generate three-dimensional intestinal organoids from human pluripotent stem cells using an approach known as directed differentiation. In October 2011, Dr. Spence joined the faculty of the University of Michigan Medical School. His lab is currently pursuing projects to understand intestinal development and function, and also studies lung development using mice and a new three-dimensional human lung organoid model the lab has recently described. Dr. Spence?
Thank you very much. Thanks for the kind introduction and the invitation to speak at this webinar. Yes, I'll start by giving a brief outline of what I'd like to talk about today. As you had mentioned in your introduction, I will start by kind of talking about some of the developmental principles that we use to think about how we want to go about using pluripotent stem cells to generate three-dimensional tissues in a dish. I will then discuss what we've been doing, kind of how to utilize these models, and so what we've been thinking about using them for and have been using them for in order to study various aspects of disease in vitro. And then I'll finish with just kind of a brief primer on what we think that these systems can be used for, so kind of the potential of the system. Okay, so to begin with, I'll start by saying that there are really two major sources of human tissues that we grow in vitro or in the tissue culture dish. And so shown by this schematic, you can see that we can start with pluripotent stem cells and use directed differentiation, which I'll talk about a little bit more in future slides, in order to generate these three-dimensional tissues that we call organoids. We can also start with tissue derived from a patient. So for example, in the schematic, it's showing the intestinal epithelium, a crypt, which houses the stem cells in the intestine, can be taken out from a patient through a biopsy, for example, and can then be grown in culture. And so for the purposes of today's seminar, this webinar, I'm really going to talk about pluripotent stem cell derived tissues that we generate in vitro and then utilize for our experiments. And so I think many of you are probably familiar, but just as a brief primer, human pluripotent stem cells is kind of a catch-all phrase that I use to refer to both embryonic stem cells and induced pluripotent stem cells. And so as the names may imply, the human embryonic stem cells are derived from the inner cell mass of a human embryo. As you can see on the top, a human embryo labeled with ICM as the inner cell mass. And induced pluripotent stem cells are derived from adult somatic cells. So here I'm showing an image of skin fibroblasts or skin cells that are grown, and they can be transduced with reprogramming factors that essentially allow them to go back in developmental time and behave like embryonic stem cells. And so for all intents and purposes, we think about these two pluripotent stem cell systems as equivalent. They have the capability for unlimited self-renewal. And then in theory, we should be able to take these pluripotent stem cells and, given the correct set of conditions, we should be able to differentiate them into any cell or tissue type in the human body. And once we've been able to differentiate or direct the differentiation into a specific cell type or tissue, we think that we can then use these tissues to model human development and disease. There are already clinical trials going on using pancreatic beta-like cells that have been derived from embryonic stem cells, and so these tissues are, again, theoretically and practically useful for transplantation. And we think then that we could also use this tissue for tissue-specific drug screens or toxicity testing. Okay, so in thinking about how we are starting with this undifferentiated pluripotent cell that can give rise to any tissue in the body, the real question that we like to ask as developmental biologists is, how do we go from this undifferentiated state to a lineage-specific to induce lineage-specific differentiation? So, for example, if we want to make intestinal tissue, how do we go from point A to point B? And the approach that we take is by going back and really thinking about how the embryo would do this. And so we have decades of information from studies using different model organisms, such as frogs, fish, mice, and chick embryos. And so these systems that have been employed for decades, and in fact hundreds of years, have really informed our understanding of embryonic development. And so we like to go back and take what we can from that information and then apply it in the dish to think about how to really inform our studies. And so what I'm showing you here is a schematic of early mouse development. And so on the top is kind of this developmental progression from being just an embryo to being an early, to then being kind of an early organized embryo, and then kind of a later stage embryo where you can see clear differentiation and clear domains. And so in the center picture, I'm showing you an embryo just post-gastrulation, and pseudo-colored in yellow is the endoderm. And there's a little cartoon of the endoderm below that middle panel where you see the A and the P representing anterior and posterior, or front and back of the embryo. And so this endoderm is really going to give rise to the lining of your entire GI tract, so your esophagus, the lining of your stomach, the lining of your intestines, as well as the associated organs, the liver, pancreas, lungs, etc. And so really we start by saying, how does the embryo, how does this process happen in the embryo? And so I should also mention that this kind of flat sheet or this cup-shaped sheet shown in the center here really undergoes these three-dimensional changes over time as well, giving rise to these different domains, which I've marked with foregut, midgut, and hindgut, or kind of FG, MG, and HG. And so those different domains give rise to different structures within the adult body plan. And so each one of these domains, the foregut, midgut, and hindgut, are kind of molecularly distinct very early in development, and then, as you can see in the schematic, give rise to different subsets of organs. And so the general idea in thinking about how we use this information to then generate tissues in a dish from pluripotent stem cells is that we really want to understand or take what we can from these, what we know from embryonic development, and then apply that into the dish. And so here's kind of a schematic of decades worth of work that really has informed us that this signaling molecule called activin A, which stimulates TGF-beta signaling and mimics kind of nodal signaling in the embryo, is able to induce gastrulation into an endodermal lineage, and that then there are patterning factors. So once you have endoderm made, that in order to get these different molecularly distinct domains along the embryonic axis, you have to have, for example, inhibitors of certain pathways on one side of the embryo and activators of different pathways on the other side of the embryo. So I'm just showing a couple of examples of signaling pathway activators and inhibitors that might be helping to pattern this tissue into different molecular domains. So this is now just to point out many of this work going back several decades now that has really kind of blazed the trail for our ability to do this in this dish. Okay, so this is a summary of several published studies, both by my lab when I was a postdoc in Cincinnati and by others, showing that we've really been able to take these different embryonic cues and kind of mimic development in a stepwise fashion, whereby you start with an undifferentiated pluripotent stem cell, and you can treat it with activin A to generate endoderm, shown in the middle panel with the kind of green-blue cells. And then we can further pattern this endoderm. So you can think about this endoderm as kind of a naive sheet of cells on a tissue culture dish that has the capability to give rise to any of those different endodermally derived organs. And so we found that by, for example, activating FGF4 and Wnt3A to get by activating FGF and Wnt signaling together, we can really instruct these cells to become intestine. Others have shown that by inhibiting BMP signaling and TGF-beta signaling, you can drive these cells into a foregut lineage, and further, you can then take these foregut cells and differentiate them into stomach or lung tissue. So one of the things that we noticed is that when we were generating these, attempting to generate intestinal tissue from these pluripotent stem cells, that in addition to simply turning on transcription factors that are expressed in the intestine, we also saw the kind of the appearance of these three-dimensional structures in the tissue culture dish. And so you can see here in the right-hand panel cells, endoderm cells treated with FGF4 and Wnt3A have these tube-like structures denoted by the yellow arrowheads and these 3D floating aggregates denoted by the black arrowheads. And in contrast, if you have endoderm and don't treat with any growth factors as a control, you never see these three-dimensional structures arising in the dish. And so what we were able to do then is to take these clusters and put them in a three-dimensional matrix. As Dr. Kim had discussed in his talk, Matrigel is a three-dimensional extracellular matrix composed of several matrix components. It forms a gel. So you can kind of think about each one of these little spots at day 0 here as one of these little three-dimensional clusters suspended in three-dimensional space. And then by growing them or adding the right combination of growth factors, we were able to expand these small little aggregates of cells into much larger structures that we call organoids. And so shown in the bottom is a picture of an organoid that's been grown for about a month in culture. And so you can see it essentially looks like a soccer ball. So it's a large hollow ball. It's made up of an epithelium with a fluid-filled lumen, and it's surrounded, as you can see, the kind of fuzzy, nondescript looking tissue towards the peripheral of this ball. It has a mesenchymal layer, so it has supporting cells adjacent to the epithelium. And we were able to show that these organoids possess all of the major cell types of the small intestine, and so we call them human intestinal organoids, or you may see me refer to them as HIOs for short. But in essence, at the bottom, there's a cartoon of the organization of the intestine. So at the bottom, those yellow cells are Paneth cells, and interspersed amongst the Paneth cells are stem cells, which proliferate and give rise to all of the different tissue lineages of the small intestine. So I'm showing you 4 of those lineages here: enterocytes, enteroendocrine cells, Paneth cells, and goblet cells that are present in the intestinal organoids. Okay, so at this point we have human intestinal organoids. We're able to direct differentiation of pluripotent stem cells into these 3D tissues using developmental principles. And so the question is, what can we do with them? And those kind of, we're still asking ourselves those kind of questions and always trying to think of new ways that we can apply these to understand development or disease in the lab. But I'm going to talk to you about one story where we have been looking at epithelial barrier function. And so the intestinal epithelium is quite unique because it has a dual role where it has to both absorb nutrients and it also has to perform a protective barrier against the food that we eat and against the bacteria that live in our gut. And so the way that it does this is that it forms a tight physical barrier called, which is formed by these epithelial junctions, shown here in this cartoon. And you can see the tight junctions kind of marked by the claudin 2 and then the red and the blue proteins. And so these are junctions or adhesive junctions between two cells that really hold them in really close proximity. And they prevent things like dietary antigens and bacteria from moving across the epithelium. And in the event that these tight junctions get disrupted, things can move across the epithelium. They get sensed by the underlying immune system and can start an inflammatory cascade. And so this tight epithelial barrier is really critical for normal homeostasis of the gut. And so one of the questions we wanted to ask is whether the organoids had tight epithelial junctions and whether they had barrier function. And so when we just looked at the epithelium of these organoids, we could see that they had the right machinery to have an epithelial barrier that was functional. And so you can see ZO1, which is a tight junction staining towards the top of this panel. You can also see E-cadherin, which is important for adherens junctions for two cells that are adjacent to one another to form a junction. And so this epithelium in these organoids really looked like it should have a functional epithelial barrier, but we really wanted to design an assay where we could test the function of this barrier. And so we used, this is a cartoon schematic of an assay that we use that has been used by others and both in vitro and in vivo, whereby if you think about one of these organoids, as I had said, as kind of a fluid-filled ball, and so the organoid is represented by this orange, this yellow and orange structure, with the yellow being the inner epithelial layer. And so what we can do is we can simply add a molecule called physiodextran of 4 kilodaltons in size into the media. And what we know is that normally, if the epithelial barrier is intact, the epithelial junctions keep this molecule out. And so the molecule is big enough that it can't penetrate through the epithelial layer unless the tight junctions are injured. And so in the case of a disrupted intestinal barrier, we would see this green fluorescent molecule travel into the lumen of the organoid and get trapped. And so as kind of a proof of concept, we incubated our organoids in physiodextran for an hour. And so those are the two panels on the kind of leftmost and the second, so the one adjacent to that. And then, or we incubated them with physiodextran plus EGTA. And so the EGTA is designed to break down the epithelial barriers. And so what you can see is that in controls, the organoids really nicely keep this dye from moving into the center of the organoid. And I should say that under the bright field images, each one of those structures is an individual organoid. And so then on the right, you can see when we add a chemical that disrupts the barrier, that we can see a nice movement of this physiodextran dye into the center of the organoid. So this really tells us that using this chemical, that we're able to injure the epithelial barrier, disrupt the tight junctions, and that the barrier is now allowing dye to flow into the lumen of the organoid. And so we also saw, when we looked histologically at these organoids, that the loss of barrier function is associated with dynamic changes in adhesion proteins. And so we're looking at E-cadherin here, which is normally on the top in the control, found on the basal and lateral surfaces of the cells, and they kind of line up like this nice set of these columns. And so it's a columnar epithelium. And you can see that in the EGTA treated, where we disrupted the epithelial barrier, that we have a quite disorganized E-cadherin staining. So then we've been using a chemical to disrupt the epithelial barrier up to this point. What we really wanted to know was whether there were physiological sources or chemicals, physiological means that we could disrupt the barrier such that we could model disease and then move forward and study and understand epithelial barrier disruption in the context of disease better. And so in collaboration with a close colleague of mine, Vince Yeon, here at the University of Michigan, who studies Clostridium difficile, we wanted to ask whether using toxins produced by Clostridium difficile whether we could model kind of the disease pathogenesis induced by this bacteria. And so Clostridium difficile is an anaerobic spore-forming gram-positive bacteria. There are prevalent outbreaks and infections in hospitals and other facilities where people live together in close proximity. These disease rates are on the rise also in the pediatric population. And so this bacteria releases two active toxins, toxin A and toxin B. The nomenclature for toxin A is TCDA. The nomenclature for toxin B is TCDB. Toxins are a glucosal transferase, and they cause pseudomembranous colitis and also can cause death. And so the way that these toxins work, so this is kind of the experimental setup, and then I'll tell you how the toxins work. So what we've done in order to ask whether these toxins can disrupt the barrier of our intestinal organoids that we've taken an organoid and we've added one of the purified toxins, either A or B, to the organoids for 2 hours, and we've washed the organoids out, and so washed the toxin out from the organoids. So the idea is that the toxins would induce damage to the epithelium and disrupt the barriers. Then we add this fluorescent dye, physiologically, for 1 hour, see if the dye moves into the lumen of the organoid, and then we wash the dye out and we can analyze to see if the barrier has been disrupted. Okay, so the way that the toxin works, sorry, the slides are a wee bit out of order. I'm just going to skip ahead and then I'll go back. So the way that the toxin works is that they kind of decrease Rho-RAC CDC42 activity, which decreases phosphopaxillin, causing a loss of cytoskeletal integrity and leading to adhesion and polarity defects. And so what you can see here from published data from others is that in using a fibroblast cell line, NIH3T3 cells, normally these cells are kind of grow as individual cells on a plastic of a tissue culture dish, and they're kind of spread out. And you can see when you add one of these toxins that they very rapidly have cytoskeletal abnormalities, leading to the cells to round up. They'll eventually delaminate from the tissue culture dish and die. Okay, so back. So what happens when we add these toxins to the organoids? This is again a control, or this is a control, both a negative control and a positive control. So this is the same experiment that I showed you before, using where we add nothing, using no disruption of epithelial barrier function. If we add the chemical A, we can then see a nice robust reduction of barrier loss. So we've injured the barrier in this case. If we add TCDA, so toxin A, at 2 different concentrations, again, remember that green means epithelial barrier is disrupted. So we can see at low concentrations at the top that the barrier really seems to still be maintained, but at higher concentrations, have a complete disruption of the barrier in all of the organoids in this experiment, in which case you can see 13 out of 13 had a disrupted barrier. When we looked at toxin B, again using 2 different concentrations of toxin, we can see that some of the organoids have an epithelial barrier that's disrupted, but that it's a small number, a small percentage compared to that's toxin A treated. And so again, just to remind you, this is kind of how we think, this is how we understand that the toxins work. And since it appeared to us that toxin A was more potent or was induced more robust epithelial barrier damage, we really wanted to look to see if toxin A seemed to be acting more potently in the organoids. And what we were able to see is that when we looked at toxin A or toxin B just at the highest doses relative to an untreated control, you could see that phosphopaxillin was really reduced in the toxin A treated organoids compared to the toxin B treated organoids. And so we really did see this pretty dramatic differential response to different toxins. And we still don't really understand why one toxin acts more potently than the other, and those kind of studies are ongoing. So we were also then able to confirm kind of this functional observation. So again, remember, toxin A seemed to be more damaging than toxin B, and so we were able to confirm when we looked at the cells that seemed to be the case at the histological level and at the immunohistochemical level as well. So again, showing you adherence staining, which marks these nice adherence junctions, control, and this nice columnar epithelium. You can see that in the toxin B treated that this is somewhat disrupted, but really in the toxin A treated we saw a really dramatic shift of where the adherin was being expressed, and this really correlated with the level of damage that was done to the tissue. We looked at additional cell junctional proteins. So we looked at occludin and ZO1, both of which are important for cell-cell adhesion and for tight junctions. And so again, consistent with our functional data, for example, looking at ZO1, we really didn't see that much of a difference in the apical tight junction marker ZO1. But when we looked in the toxin A treated organoids at the bottom, we could see a really dramatic kind of redistribution of this protein so that it really wasn't present at the top of the cells in those nice punctate spots. So just to summarize this section, really what our goal was to determine whether these organoids would be useful in functional assays. And so we've been able to show then that the organoids have a functional paracellular barrier, and that different Clostridium difficile toxins have differential effects on the barrier function of the organoids, such that in our hands, CDA seems to be more toxic than TCDB, and so this poses the question, and as I had said, this is kind of ongoing research, and we still don't know the answer, but what are the different mechanisms by which these toxins are eliciting their effects on the epithelium? Okay, so just in the last couple of minutes, I've just got a few more slides to kind of talk about where we're going with these organoids in culture. And so really we look at these, and I think I've probably given you some indication that these organoid systems really can be modular systems to build biological complexity and study increasingly complex biological problems, or to study really kind of reductionist and simple biological problems as well. And so what I mean by that is that ultimately what we think we can do is, in addition to having these organoids in a dish and being able to add chemicals or purified toxins to them, that we want to be able to do things like introduce bacteria in the lumen of these organoids, similar to the bacteria that would live in your gut. So either commensal bacteria or pathogenic bacteria, to understand how your gut health and function might be influenced by different bacteria. And then shown on the outside are a couple of different cell types that are present in the normal intestine, but that are lacking from our system in the dish. So you can see an immune cell on the right-hand side and a nerve cell on the left-hand side. And so we think that we can continue to build increasing biological complexity by adding like immune cells into the dish to understand this, how the immune cells might influence the epithelium, or how an immune cell might respond to a bacteria within the lumen of this organoid. Start understanding this kind of crosstalk between different tissues, different cell types. And so I'll just show you a couple of, and then I should say, again, this is kind of a pie-in-the-sky type of kind of dream for this system, but that we might also be able to integrate flow into these organoids and out of these organoids. So we could kind of mimic the natural flow of things through your intestine. And so we could put in things like, again, pathogens, bacteria, or viruses, and then really be able to measure physiological responses like water secretion, mimicking diarrheal disease, and things of that nature. Okay, so the other thing I'd like to point out is that with the data that I showed you with Clostridium difficile, and actually going back to that schematic cartoon I just showed you with being able to inject bacteria into the middle of these organoids, is that this Clostridium difficile toxin-mediated barrier injury that I had already showed you was really opposite of what would happen in the normal gut. So of course, normally in your intestine, the bacteria would live inside the intestine, would secrete toxins, would then talk to the epithelium and damage it. And the experiment that we were doing, as you can see on the left, was really to put the purified toxins on the outside. So the natural situation is kind of on the right-hand side of this cartoon, and the artificial situation that I talked about previously is on the left-hand side. So what we really wanted to know was whether this kind of differential exposure of the lumen or the apical epithelial surface to the toxins was somehow responsible for these different responses that we were seeing. And this is also as a start towards building up to being able to inject things like bacteria into the organoids. And so we really wanted to refine our assays and develop a new assay where we could micro-inject things like toxins and this fluorescent dye into the middle of these organoids. And so we showed that we were able to kind of recapitulate the results of just dumping these toxins on the outside of the organoids, in that the control organoids really over time seem to maintain nice. So now it's the opposite way around. I should mention that if the barrier is damaged, the fluorescence should leak out. So the green color should leak out and fade away in the absence of an injury to the epithelium. So control, you can see that it maintains the fluorescence. When we add the toxin A, we see a pretty quick loss of fluorescence. The toxin B, we still see somewhat of a reduction, but it's not statistically significant compared to control, and it's not nearly as significant as compared to toxin A. So this really correlated well with the results that we had got by just dumping toxins into the media, and we can quantitate the loss of fluorescence over time, shown in this graph here. So you can see that the blue and red are the control and the TCB-treated organoids, and they really lose a little bit of fluorescence intensity, but not as much as compared to the toxin A, which is statistically significantly more. Okay, so then moving forward, again, we really wanted to ask whether we could microinject live bacteria into these organoids and really start to model these more complex bacterial host interactions. So we started as a first pass by doing a proof of principle experiment where we injected E. coli. So in this case, this is a commensal form of E. coli that was isolated from a human intestine. It's been labeled with GFP, and so you can see each one of those little spots is a bacterium associated with the orange epithelium of the organoid, and we've been able to co-culture these microbes within the organoids for up to 5 days at this point. And so we went back and said, well, if we add the Clostridium difficile into the organoids, can we model this same barrier disruption? So we used 2 strains of Clostridium difficile, a toxigenic strain that secretes both toxin A and toxin B, and a non-toxigenic strain, so one that doesn't secrete the toxins. And we can see in the histological sections of the non-toxigenic Clostridium difficile a really nice epithelium shown here, and you can see that purple haze in the center of panel C is the bacteria. In contrast, if you look at panel B, you can see that the bacteria are present, but the epithelium is completely gone. So this epithelium has, at this point, after about 12 hours in culture, already undergone such severe injury that it's completely, the cells have completely lost their adhesion to one another. They've rounded up and sloughed off and have undergone cell death. And so, again, we can see the conclusion here is that injected Clostridium difficile also induces a massive damage to this epithelium. And similar to, so again, this is just to show you a time course of the purified toxin compared to the Clostridium difficile toxigenic bacteria itself. And so in each one of these panels are organoids that have been injected with TCA and the phytidextran or Clostridium difficile, and the phytidextran, and this is a time course really looking at the injury over time. So with that, I will wrap up and give you some conclusions. Hopefully I've been able to give you some insights into the way that we think about generating these three-dimensional tissues, and also how we're thinking about applying these in the lab to study development and disease. The data I've shown you today has really been designed to stress the point that organoids, human intestinal organoids, have a functional epithelial barrier, that we can use various means, chemical or from Clostridium difficile purified toxins, that to disrupt this barrier, and that the toxins actually have different abilities to disrupt the barrier, suggesting that there's something different about the way that these toxins act on the epithelium. It seems that both apical and basal exposure to the toxin disrupts the barrier. Intestinal organoids seem to be ideal for delivery of luminal contents via microinjection, and so those were the studies with the bacteria I showed you at the end, and being able to inject the phytidextran into the center of these organoids. And so really, again, one of the goals for the long term is to figure out how to engineer these organoids with micro or mini fluidics, create luminal flow for long-term culture of live bacteria organoid co-cultures. We think that these, we know that these organoid microbe and organoid immune co-cultures, I didn't show any of the immune data today, are robust and stable for many days. They allow us kind of for modular assembly of increasing biological complexity in order to study various questions. So with that, I will thank, of course, the people that did the work. This is my lab as of about a year ago. The people that were really involved in the work that I presented today were some very talented people in the lab. Sha Huang and David Hill really led the Clostridium difficile and barrier function studies, as well as my group of really great collaborators, both at the University of Michigan and beyond. And of course, I thank my funding source. Thanks very much.
Thank you, Dr. Spence. Quite a few questions have come in, so let's get started with our Q&A session. This question looks like it could be answered by both of you or either of you, so you can have at it. In both of these 3D culture models, how reproducible are the results? How many organoids or matrigel preps do you need to examine to account for variation in cell types and responses?
I can start first. This is Yeon Kim, and this 3D culture matrix was quite reproducible. It's like it doesn't require those robust, like high complicated structures. So the culture models we've been watching is very consistent in the time, and different cell lines actually have a different traits. But in the same cell line, we have very similar the age of the A beta aggregation and very similar time, and we see the top pathologies. So it's quite reproducible in our hands.
Yeah, I think that's a really great question that applies not just to the organoids, but to any experimental design and experimental result. For us, the way that we design the experiments is that we try to reduce individual variability from individual organoids for each experiment by pooling organoids together for our analysis, and also by running many biological replicates. And so we often will run at least 3, but more likely or more usually on the order of 6 independent biological replicates. And by biological replicate, what I mean by that is that we might make the organoids, or the organoids might be generated from the same cell line, but we run the experiment on different days. So usually we're running an experiment 3 times from the same cell line with, and then kind of pooling all of that data together to get one result. And within each time we run the experiment, we're then having statistical replicates. So we're running 3 wells of pooled organoids. And so that way we hope to reduce what might be a strange result that's just from one organoid. We're also concerned with robustness across cell lines, and so we often will validate our major findings. If we tend to have our favorite cell line that we like to use, so we'll often validate our major finding with 1 or 2 additional cell lines just to make sure that it's a robust result.
Can I add one more thing in my side?
Absolutely.
Sure. So one of the fluctuation or those changes we observed is because our cell line is in nature is mixed culture. So we have actually see passage number variation because depending on the passages, cell line start expressing low amount of A beta, and we see that aggregation of A beta and tau is actually delayed. So what we are trying to do since then is that we're trying to make a single clonal line. And actually we have a lot of the improvement in terms of reproducibility because the single cell line is more consistent and homogeneous.
Great, thank you. This question is for Dr. Spence. How physiologically relevant are the amounts of toxins you're adding to the organoids? Is 400 nanograms per milliliter what a cell would see in vivo?
Well, it's a really great question. I think it's difficult to really answer that with a high degree of confidence. I think that we think that we're in a physiological range, so we're not using super physiologic doses. However, it's also very difficult to measure how much toxin is present in vivo since it's such a dynamic environment. But certainly, this is what I think what most people would consider to be somewhere in the range of physiologic dose.
Thank you. Dr. Spence, this one's for you as well. How self-healing are the barriers? You've shown that you can inject and monitor compartment variables. Do the barriers leak after a certain number of injections, or are they able to heal?
Yeah, that's a super question, and it's one that we have not actually addressed experimentally. So, I mean, I think that we could think about ways to do a really nicely controlled experiment to look at that, but we haven't actually done it. It's a really good idea.
Thank you. This one is for Dr. Kim. You've demonstrated that you can recapitulate amyloid and tau aggregation, but have you seen evidence in your cultures of cell death and neurodegeneration?
Actually, answer is yes, but we are very cautious because we definitely observed that additional, like more robust neuronal death in the AD cell line as compared to the control line. But as you know, that I think most important thing is whether these cell death is really relevant to the AD pathology over there, and that part we are working very hard on it now.
Thank you. This one's for Dr. Kim as well. What do you suspect is the cause of the lack of oligodendrocytes in your cultures? Would there be a benefit to encouraging their growth?
That's a very good question. So, yeah, maybe definitely I can say that our cells in our 3D culture doesn't have any myelinations, so that's not the natural conditions. But I think it's very hard to answer at this moment. Yeah, it definitely is different from the brain conditions, but we don't really know whether that leads to more robust pathology or less. That part is that we need more study to answer.
Thank you, Dr. Kim. This question is for Dr. Spence. Is it known whether the different strains of C. diff that are spread throughout care facilities with various antibiotic resistance, if they produce more toxins than other strains?
Yes, so there are definitely a lot of variations between different strains. Now, I will also make a caveat here and say that I don't actually study C. diff other than really to use it as a model for barrier injury in our system. But my colleague that I do these studies in collaboration with is an infectious disease physician and scientist, and so through my discussions with him, it is quite apparent that different strains express different levels of toxin A and toxin B, and then if you look at different strains, they will also make different levels of toxin in a strain-dependent manner.
Great, thank you. Question is for Dr. Kim. Were any of if you're familiar with any of the new and emerging metal attenuating compounds tested in the dish in this assay? If so, were they found to be effective in clearing A-beta plaques and subsequently clearing the tau tangles?
Yes, I'm very interested in because our close collaborator, Dr. Tanji's group, and also the other groups is working on that. Yes, we have a plan, and then we actually have some projects going on right now. And the idea there is that what we shown was that when we block A-beta generation, that leads to those decrease of tangle and all the things. But I think that the most interesting question is whether if we block the A-beta aggregation by the metal chelators, others, and then may lead to same effects or not. And that's actually, we are working on it now.
Thank you. Dr. Kim, what types of experimental differences did you see in the different cell lines that you tested? You tested the cell lines that you could purchase, and you tested iPSCs. What were the differences that you found?
Okay, so yeah, we actually tested several lines, and then one of the technical problems we have is when you overexpress our FAD genes. And most of cell line, including iPSC-derived human progenitor cells and several lines, they just died. It's somehow the Rancell VM we use, actually they are very strong in the progenitor conditions. They don't actually affected by the high expression of those older genes. And then they show some toxicity after differentiation. But for all the other cell line we see, there are huge problems with the toxicity, and that's the observation we had. So that at this moment, we are actually working on Rancell and several others, but Rancell is the best model.
Great, thank you. We have time now for just one more question, and it is for both of you. So again, feel free to take turns. Are there any special considerations or precautions to take when analyzing cultures with multiple cell types? How does it impact your data analysis?
So I can answer first. So I think that it does definitely impact the analysis, and so I think you have to think about it. It might depend on, or it might alter what type of analysis you choose to do. For example, if you, in our system, if you use Western blot or qRT-PCR, often we're doing it on a pool of, we're doing the assay on a pool of cells, and so we have to keep in mind that we're really looking at the response, the kind of global response within the organoid, and so we can't make too, I think we just have to caution our interpretation of the data. And if we really want to, we see an effect and we think it might really be predominantly happening in one cell within the population of cells, we really then have to go back in and look at a single cell level on the response in the organ. And so I think those are the kind of caveats. The other thing that we often consider or practically do in the lab is to think about how we can take the different cell types apart and purify them individually in order to just look at one cell, the response of one cell type amongst the community of cells. So yeah, it's definitely it's an important point and a really important consideration experimentally.
So yeah, I completely agree. And one more thing I would like to add in our side is that because it differentiates into neuron and astrocyte and several other cell types. So yeah, we've been doing those quantitative RT-PCR and several marker staining, but that's always a concern because the changes actually came not only from A beta, but the altered differentiation or fate. And that part is actually, we are actively working on it at this moment. At least for the ran cells, we don't see any big differences. That's why we think this system is working. But for the other system like hPSCS, the overexpression of these FAD is not just to kill the cells, actually it affects cellular status. And that is actually very challenging, and I think that we need a lot of progress in the future in that side. And one more thing I want to add is that even the complicated system, the change of different cell type might be advantageous. So let's say that we have astrocytes and neurons in our system, and as you well know that in the Alzheimer's brains, it's not only neuron just dies. Astrocytes activate because of that, and microglia activates. So that's another thing that we are hoping to do, that we make a same differentiation, exactly same, and then express those pathogenic conditions in the one exact same condition, and then look at impacts on other than neuronal cells and how they can interact each other. That's a kind of goal in the future.
Great. Thank you both. Unfortunately, that's all we have time for today. If you have any other questions, please consider reaching out to the speakers directly. Their email addresses are displayed on the screen. As a reminder, the webinar will be archived on the Scientist website, and you'll receive an email notifying you when the on-demand webinar is available. I'd like to thank everyone who took the time to join us today and for sharing your questions and comments. On behalf of the Scientists, I'd also like to thank our speakers, Dr. Kim and Dr. Spence, as well as our webinar sponsors, Rockland Immunochemicals and Baker Ruskin. Thank you, everyone, and goodbye.